So simulating tissues is challenging because in other modeling approaches, what you are actually simulating a gene regulatory network or things like that, objects are actually they don't have a physical dimension. But when you are actually simulating a tissue, you need to simulate cells as physical objects that actually occupy some space. That is something that is at the beginning is challenging. On the other hand, cells are not isolated objects. And when you are simulating a tissue, what we need to do is actually to simulate also the cellular interactions. And those go from paracrine signaling, so long distance signaling, because you can have diffusive factors that actually making like morphogenes cells to communicate. Or you can actually also want to simulate just justacrine interactions, like when you have a ligand and receptor in neighboring cells. The other thing that makes simulating tissues challenging is the cellular mechanics. It's not just that the cells are actually objects with a physical dimension, but actually they can deform. And that is something that needs to be taken into account. And the other thing is tissue plasticity. Cells are not objects that remains in a given place in space, but actually they can interchange their neighbors, they can actually disappear, and that is also something to simulate. Cells can grow and cells can divide. That is another thing that must be taken into account. From the computational viewpoint, another thing that is challenging is that there are many different or distinct time scales. You have to simulate things that goes from a scale of seconds or even less than that, like signaling processes, to days, because the cell cycle and the division or the growth can actually be in that scale. And last but not least, you have to integrate all this with DNA, a synthetic DNA, so gene regulatory networks, or as you want to put it. But depending on how the cells communicate or depending on their position in a tissue, they can actually lead to different expression profiles. So now all this makes simulating tissues very challenging. Now, as for simulating tissues, there are many different approaches that has been proposed, and depending on the level of detail that we want to simulate, those goes from continuous models, where actually mostly what you do is actually to simulate the tissue. I'm going to be a little bit obscure here, or mathematical here, like a field, a scalar field, so something that changes continuously in space. In that approximation, actually, you can simulate the tissues, but you can actually lose the level of microscopic detail. And on the other scale, you can actually go to models where you can have a subcellular description of the cells, like the cellular pods model. In the middle, there are other intermediate approximations that you can actually choose for simulating tissues. I'm going to just briefly discuss two models that actually I am familiar or more familiar with, that are the cellular pods model or the vertex model. And something that I like about the vertex model is that as you can see, it's in the middle between actually the subcellular and the supercellular approximation or level of detail. So that is actually good in order, because it's actually very useful, because you can describe many different situations with this kind of approximation. But let me first talk a little bit about the cellular pods model. So the cellular pods model actually describes tissues in a lattice. And a given cell, like the green cell that you can actually see here, is actually composed by a different number of pixels. What is important about the formula that you have on the right is that what you do is actually you provide for each of those pixels a given energy, and that energy actually takes into account that the cells or that pixel belong to a particular cell and the interactions between the different pixels such that you can actually conserve a given area or a given perimeter for those cells. This is a very crude description about what is a cellular pulse model. The pros of the cellular pulse model is that they have a flexible topology, so the shapes of the cells can actually be very different. You can actually simulate what is called non-confluent modeling tissues, so that means that you can have different gaps between cells and things like that. But it has a number of cons. One of them is that it's based, as I said at the beginning, on a lattice description. And that means that you can actually have in the simulation lattice-based artifact, meaning that depending on the level of the resolution that you are actually using, the results can actually be different. And that is not good. The other thing is that it is computationally intensive, because normally one particular cell can be described by, I don't know, 100 of lattice points at least, or I would say at least if you want a good resolution. And then you can have, you need to take into account that at least you can have up to, depending on the size, of course, that you want to simulate, at least 10 to the 4 cells. So that means that actually you are simulating millions and millions of points per every time step. And that is actually very challenging. The other thing is that the mechanical interpretations about the interaction of the cells is very challenging also, because many times when you have a tissue, you actually are able to measure forces or these kind of things. But here you don't have forces. You have provided an energy to a particular pixel, and actually approximation from that to the cells or the forces that are exerted by cell is actually difficult. And last but not least, there is time is difficult to actually to have an interpretation, because all these schemes based on the cellular core models are based on what is called the metropolis algorithm. And that means that time is not really a continuous quantity. Well, it never is in computational models. But in this case, how times correlates with what is called a Monte Carlo step is also challenging, particularly because, as I said before, you want to integrate different time scales. Okay. But besides all these cons, but also the pros, this is one particular simulation that I like. You can actually look for this movie if you Google in YouTube Johan's Textor, the one that you have, and there is a lecture online about this. There is actually the CompuCell and CompuCell 3D is a free software that you can actually use for simulating tissues. Using it on the cellular pulse model. And this is how a simulation looks like. Oopsie. This is actually very nice because you can actually have what is called a confluent tissue, the tissue that is in the middle, or the gray cells, if you want, in the middle and outside, that where all the cells are actually stick together. But actually, you can have also cells that actually are traveling around with not very well defined shapes that actually migrate. And this is actually simulating the interaction of, this is cancer cells, if I remember well, and macrophages. Okay. Another approximation that is the one that I am going to focus mostly on this talk and the tools, it is called as the vertex model. So the vertex model was initially thought to describe epithelial tissues. So as you can see on the top left, cells are actually epithelial cells are polarized. That means that mostly all the mechanical interaction are happening on the what is called the apical plane, the cells. That means that mostly, if you want to describe the dynamics of the cell, you can actually focus on that plane. And, and given that description on the optical size, the vertex model, what it does is instead of describing cells like a group of pixels, it's actually a lattice-free modeling approximation. What you can see is that the epithelial cells also mostly what they have is a polygonal shape. And this vertex model, what it does is simulate the vertices of these polygons. So that means that one of the main advantages of the vertex model with respect to the cellular pulse model is that, as I said before, when you have the cellular pulse model, you need many, many lattice points. And actually, you need a lattice. And in the vertex approach, you can describe one cell with a limited, very limited number of points that are the vertices, and it's lattice-free. So this, because of the computational advantages of this model, it has progressed a lot during the last years, and this is the tools that I'm going to present today are actually based on the vertex model. The vertex model approach actually shows you that you can actually easily implement what is called the tissue plasticity through a series of transitions that are called the T1 transitions, where you have neighbor interchange or cellular extrusion, and cellular division can be actually implemented. I'm not going to say easy, but it can be easily implemented somehow with respect to other. Something that has been seen in real tissues that is something that can actually simulate the vertex model is that there is actually, as a function of the mechanical parameters, and here forces are, as you will see, are actually something that you can actually measure and correlate with the behavior of tissues, is the behavior of the vertex model depending on the different parameters that you have, that you can actually lead to different behaviors. What is called a state where the cortical tension of the cells dominates the behavior of the tissue, and another particular profile of the tissues, that is where adhesion actually dominates the behavior of the tissue. And this actually has been seen in real tissues, and that is something that can simulate the vertex. To go a little bit into details, as I said, what you do with the vertex model is that you simulate the behavior of the vertices of different cells. So in this case, in the image that you have on the screen, if you want to describe what is the behavior of that particular vertex with index I, with a given position R sub I, what you provide to that vertex is an energy that takes into account the elasticity. So that means that the cell will like to have a particular area. The tension, that means that they have a particular affinity depending on the cells and in the environment, and also the contractility that forces the cell to have a particular perimeter, and this is related with the cortex activity. In that publication that you have on the bottom, we describe different approaches for simulating actually tissues and their organization, if you want further details. Okay. Something that can be actually, in this particular paper, is one of the papers, not the first at all, but one of the papers that were more successful for demonstrating that actually the vertex model can simulate the behavior of tissues, of real tissues. It's a wonderful paper in terms of modeling from the organization of the tissues and then different conditions, the different polygonal organization, the mechanical properties, and even something that is shown below, that is actually when you perform a laser ablation between two different cells to see how they expand. And this is very important in order to measure different parameters that have to do with the mechanics. So if you want to learn a little bit more about the vertex model in a real context of or successful context for simulating tissues, this is a publication that I would recommend. I'm going to skip this. Okay, let's go to the details of TiFoSi and ANISE. So TiFoSi and ANISE are two tools that we developed in the past. On the bottom, you have the publications that in bioinformatics that led to these tools. And so they work as follows. We first developed TiFoSi, that is an acronym for, let me move out this that you cannot see, but I can. Okay. No. Sorry about this. I'm trying to skip one panel. Remove one panel that I can see in my screen. Okay. So TiFoSi stands for Tissues, Forces, and Signaling. And it's actually a simulation code that is able to integrate both the mechanics and all the parameters that have to do with the simulation of tissues. And it's actually written using a language that is XML code. So actually you write this with thought, and we think that is very clear for writing the code or stating your simulation. But we wanted the code to be very fast. Very fast normally means using C++. So what it does is that you write in a test file the XML code, and then there is a Python code that actually reads that file and recompiles all the source codes of C++, and then can actually run the data. Because we wanted also to be fast, it's not actually focused on providing a graphical result, but actually to providing data of all kinds about the simulation of the tissues. But with the TiFoSi, we also actually provide some visualization tools such that you can actually use that data that is generated by the code and so what is the result of your tissue. I'm going to, can you see my browser?
Yes.
Paula, okay, thank you. Thank you, Cristina. So this is actually the web page for this tool. And here you can see an overview. You can actually see different files, a wiki. You can have all kind of information. In particular, you can actually go to a visualization tools where you can actually download. You can go to examples where we go provide sample files about how to configure the XML file. We can actually, there is a very detailed manual that we developed for TiFoSi, and all kinds of information can actually be seen here. Okay, now one of the problems that, and now I am back hopefully to the presentation. So one of the problems that we found with TiFoSi is that biologists were not using TiFoSi. So we wanted this tool actually to be used by biologists in order to have a computational tool for simulating their tissues. And they were not using. One of the reasons that we found out is that even though the simulation for writing the XML code is actually very simple and detailed in the manual, how to actually set up a computational experiment for TiFoSi, they did prefer to use a visual tool for generating the XML configuration. So because of that, what we did is actually simulating or creating this other tool that is called ANISE. So ANISE, well, there is a manual also, but it's actually a web service that you can actually install in your simulation. And all the things that actually can do when you are creating the XML file can actually be done with ANISE. I'm not going to enter into details, but actually the initial configuration of your tissue, you can actually delete cell types. You can actually create a new cell type that I'm going to call, I don't know, BCB, set. And then if you want actually to set what is the initial position of these cells, you can actually draw the cells, things like that, scale the tissue, larger or smaller, whatever you want to do. Once you actually set up the initial configuration, you can actually go to different stages because maybe in your tissue you want to simulate a tissue that at some time is actually not growing and then it's actually growing and things like that. So you can actually define different stages in your simulations. Add here in the sample is actually given 2. In this stage, for instance, you can see that there is no growth of the cells. You can actually turn growth of the cells, whatever you want to do. Maybe the cell DNA is actually not really active. You can actually turn off and whatever you want to do in the different stages. For the proteins, or how you define the gene regulatory networks, there are different ways to do that, but one of the things that we developed with this is actually to do a tool that actually you can use, I'm going to call this protein BCB1. I'm going to provide a particular degradation rate, a basal rate of expression. If this protein can actually diffuse out of the cells, you can actually set it up or not. You can actually provide an initial value for that protein that can be stochastic or not. And then once you have proteins like that, let me call another protein BCB2, then you can actually create positive interactions. So this protein is actually positively regulating this protein, is actually positively regulating this. You can actually create a negative regulation that can actually go to itself, to an interaction, to the other protein. And depending on how you actually create your visual interaction between different genes or proteins, you actually create what are the modeling equations that actually you have in the system. All these interactions can actually are modeled through a Hill function. Depending on the properties that you want for the Hill function, actually you set up the model in a very graphical one. And then you have different cellular interactions. For instance, you have the cellular interactions between a particular cell type or the surrounding space. You can actually set up different mechanical parameters. You can actually set up the parameters between the different cell types, whatever you want to do. And this is actually highly configurable also in a very graphical. If you want an external force, because maybe some cells are actually want to migrate, you can actually do it in a very graphical way. And then in terms of the cell cycle, you can define what is actually the direction of the division angle, how actually what is depending on your experiments, if this is can be a very noisy one in terms of the cellular direction or not. You can actually define how the cell area is going to change as a function of time, and so on and so forth. So all these tools that is ANISE, actually all that is doing is actually just generating this XML file that I mentioned at first. So all these setup create all the different conditions in order to simulate a tissue that you can actually use, hopefully in a very easy way. I'm going to skip this, but I just want to say that the approximation that we use in order to simulate gene interactions or protein interaction is based in this paper, where we demonstrated that you can actually go from a graphical representation to a well-defined mathematical representation, and that can actually reproduce all kind of logical gates or whatever. So I'm going to show you to finish just some examples where we use this approximation for simulating tissues. In this paper, what we did is actually simulating something that is a problem in developmental biology. That is, when you actually have a tissue that is actually growing, but you want to separate different domains of the tissue. There are many examples during development where and when you want to do this. This is actually this particular paper is based on the development of the wing disc or the wing of the flight, but it's actually that applies to the separation of the rhombomeres during vertebrate development. So in this case, what we want to understand better what were the mechanical interactions between the different cellular populations and one particular population, that is the red one, that is actually what is called the organizer, that is able to separate cells of these two domains and avoid intermingling between the cells. So we actually, as the other cells are growing, actually this is just an elongation of that particular cellular population. And at the same time, it's actually an active barrier to avoid the migration between one compartment and the other. So we demonstrated that actually what were the mechanical interactions that were required in order to do that. We demonstrated that particular cellular population that is separating the domains is actually strong in order to avoid the migration to cells, even they're actually pushing against the other population. I think that this is simulation now. A movie, I meant. Once that we actually were able to reproduce the behavior that was found in the wild type, one of the nice things about these tools is now you can actually ask what would be the behavior under different conditions or different mutants. So if you have actually reduced addition between different cells, that is something that can be achieved. You can actually see that or explore what are actually the limits in order to avoid or actually the robustness, if you want, of the population separating the different domains. Or you can actually have simulations where you can actually test, well, what is the role of the different cellular divisions as the tissue is growing? And actually, you can actually extrapolate that to real experiment. Okay. Another example that I'm going to provide is related with the formation of the limb, so your hands. This is a very interesting problem because there is a gene regulatory network that actually provides a particular pattern. As you can see here, the pattern of SOX9 is not equal during limb development. But actually, that pattern is very important because that is going to tell the tissue what cells are going to be a finger and what are the cells that must die through an apoptotic process. So that is actually the process that creates your hand. And this, the formation of that particular pattern is actually correlates with the elongation of the limb in one particular direction. So we wanted to understand better how actually the patterning and the signaling, if you want, and the mechanical properties actually feedback each other in such a way that you can have a maintenance of the pattern and at the same time an elongation of the tissue. This is one particular simulation where you can actually see how TiFoSi behaves, and you can have the formation of tissue. The tissue is actually growing, and the pattern is actually maintained as a function of time. There is something interesting here that you cannot see given because there is a lot of information in the simulation. You can see cells growing, dividing. But actually, when you analyze, and that is something that was shown in real tissues, what is the elongation and the division of the cells? Are the division of the cells, they're actually the elongation of the cells, they elongate in the perpendicular division to elongation of the tissue. And this was kind of a conundrum that we were able to solve using these images. Okay. And as I said before, with these tools, you can then explore, okay, now that we are able to simulate the behavior that we found in the wild type, you can actually ask different questions like what is the role of the plasticity of the tissue, the cellular divisions, what is the pattern of cell divisions as a function of space and time, and things like that. One of the last examples that we are actually working with is a collaboration in a common project, a Prometeo project with Ana Conesa, where we are actually trying to, it's a combined effort between different researchers and approaches in order to understand spinal cord injury and recovery. So this is a section of the spinal cord. You can see, you don't see there the level of details of the cells, but there are many cells over there. There is the gray and the white matter. And what we want to understand is both from spatial transcriptomics and different tools, in vivo experiments and microscopy and simulations, if we can actually understand better how or what happens during spinal cord injury and how can actually provide recovery. In this context, we are actually also using the vertex model simulation. These are preliminary results where we actually have the cellular matrix, the different types of neurons and different cell types. And we want to use these approaches with the spatial transcriptomics in order to understand cellular interactions and the behavior under injury and recovery. Okay. Last but not least, one of the other things that we are actually working now with these tools are related with tissues under stress. And I don't mean under stress, stress in the meaning that what we are playing is with stretching and compressing tissues. These are situations where tissues have to go through on the regular basis. Think in your heart when it expands and compresses. So here, what we want to understand in these kind of processes is the interaction between the mechanics and the signaling networks. In particular, we are actually simulating in these tissues what is called the Rho-myosin interactions. So in that tissue that is actually under stress, so we are actually pulling the tissue, what we can actually see is that there is this propagation of these waves of myosin that correlate with the behavior of the cellular mechanics. So this is a particular movie. The quantification of these movies can be seen on the right, where we are plotting on the vertical axis the average concentration of myosin on the cells, and on the horizontal axis we have the cellular perimeter. And you can see that there is these cycles that actually tell us that there is this that we can simulate these mechanochemical interactions. So I think that I am right now on time. I just want to thank many of the people that has been involved over the years with these sort of tools. So Uriol, that is now API in Edinburgh, was actually instrumental in order to develop these tools. And there are also collaborations with people here in Valencia that were involved in ANISE, in the graphical part. Samira, Adrian, and Victor that are, Samira is in the US now, and Adrian and Victor are PhD in the group, working on different problems using these tools. And Victor is co-supervised with Ana Conesa. Thank you for your attention. Thanks.